Biotechnology Principle and Process. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. In this sense, making curd, bread, or wine, which are all microbe mediated process, could also be taught as a form of biotechnology. However, it is used in a restricted sense today to refer to such of those processes which use genetically modified organisms to achieve the same on a large scale. Further, many other processes techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a DNA vaccine, or correcting a defective gene are all part of biotechnology. The European Federation of Biotechnology (EFB) has given a definition of biotechnology that encompasses tra- both traditional view and modern molecular biotechnology. The definition given by EFB is as follows: the integration of natural selection, no, the integration of natural sciences and organisms, cells, parts thereof, and molecular analogy of products and services. Principles of biotechnology. Among many, the two core techniques that enable both of modern biotechnology are genetic engineering, techniques to alter the chemistry of genetic material, DNA and RNA, to introduce this into host organisms and thus change the phenotype of the host organism. Second, bioprocess engineering, maintenance of sterile, microbial, combination-free ambience in chemical engineering process to enable growth of only the desired microbe eukaryotic cell in large quantities for the manufacture of biotechnology products like antibiotics, vaccines, enzymes, etc. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantage of sexual reproduction over asexual reproduction. The former provides opportunities for variation and formulation of unique combinations of genetic setup, some of which may be beneficial to organisms as well as population. Asexual reproduction preserves the genetic information, while sexual reproduction permits variation. Traditional hybridization process Traditional hybridization procedures used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with desired genes. The techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning, and gene transfer, overcome this limitation and allow us to isolate and introduce only one odd set of desirable genes without introducing undesirable gene. Introducing undesirable genes into the target organism. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, this piece of DNA would not be able to multiply itself in its progeny cells of the organism. But when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. This is because the alien piece of DNA has become part of chromosome, which has the ability to replicate. In a chromosome, there is a specific DNA sequence called the origin of replication, which is responsible for initiating replication. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be a part of chromosomes, which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template DNA. Let let us now focus on first instance of the constructions of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid, plasmid which is autonomously replicating circular extrachromosomal DNA of Salmonella typhi. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for configuring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of the so-called molecular scissors restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA acted as vectors to transfer the piece of DNA attached to it. You probably know that mosquito act as an insect vector to transfer the malarial parasite into human body. In this same way, a plasmid can be used as a vector to deliver an alien species of DNA into a host organism. The linking of antibiotic resistance gene with the plasmid vector became possible with the enzyme DNA ligase, which act on cut DNA molecules and joins their ends. This makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA, which when this DNA is transferred into Escherichia coli, which is E. coli, a bacterium closely uh, a bacterium closely related to Salmonella, Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. You can hence infer that there are three basic steps in genetically modifying an organism: identification of DNA with desirable genes, introduction of identified DNA into host, maintenance of introduced DNA in the host, and transfer of the DNA to its progeny. Tools of recombinant DNA technology. Now we know from the foregoing discussions that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools, that is restriction enzymes, polymerase enzyme, ligase, vectors, and the host organism. Let us try to understand some of this in detail. Restriction enzyme. In this year 1963, the two enzymes the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl group to DNA, while the other occurred DNA. The later, the later was called restriction endonuclease. The first, the first restriction is endonuclease HIN2, whose functioning dependent on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIN2 always cut DNA molecules at a particular point by recognizing a specific sequence of six base pairs. This specific base sequence is known as the re- recognition sequence for HIN2. Besides HIN2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequences. The convention for naming this enzyme is the first letter of this name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they were isolated. Example EcoR1, which is EcoR1, comes from Escherichia Escherichia coli RY13. In EcoR1, the letter R is derived from the name of a strain. Roman numbers following the names indicate the order the enzyme in which enzymes were isolated from that strain of bacteria. Restriction enzymes belong to a large class of enzymes called nuclease. These are two kinds, exonuclease and endonuclease. Exonuclease remove nucleotides from the ends of the DNA, whereas endonuclease makes cuts at a specific position within the DNA. Each restriction enzyme Each restriction endonuclease functioning by inspecting the length of a DNA sequence Once it finds a specific recognition sequence, it will bind to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Do you know what palindromic means? These are group of letters that form the same words when read both forward and backward. Example: Malayalam. As against a word palindromic where the same word is read in both directions, the palindrome in DNA is a sequence of base pairs that reads same on the two strands when orientation of reading is kept the same. For example, the following sequences read the same on the two strands in 5' to 3' directions. This is also true if read in the 3' to 5' direction. For example, GAATTC. When we yeah, GAATTC, restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, but between the same two bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. Figure 9.1. These are named so because they form hydrogen bonds with their complementary but counterparts. This stickiness of the ends facilitates the action of the enzyme DNA ligase. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources genomes. When cut by the same restriction enzyme, the resilient DNA fragments have the same kind of sticky ends, and this can be joined together end to end using DNA ligase. Figure 9.2. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Separation and isolation of DNA fragments. The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. And since DNA fragments are negatively charged molecules, they can be separated by forcing them to move towards the anode under an electric field through a media matrix. Nowadays, most common used matrix is agarose, which is natural polymer extracted from seaweeds. The DNA fragments separate resolve according to the science through sieving effect provided by the agarose. Hence, the smaller the fragment size, the further it moves. Look at figure 9.3 and guess at which end of the cell sample was loaded. The separated DNA fragments can be visualized only after staining the DNA with a compound known as etherium bromide, followed by exposure to UV radiation. You cannot see pure DNA fragments in the visible light, and without staining, you can sorry you can see bright orange colored bands of DNA in a etherium bromide stained gel exposed to UV light. The separated bands of DNA are cut out from the agarose ex- and extracted from the gel piece. This step is known as elution. Elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Cloning vectors. You may that plasmids and bacteriophages have the ability to replicate within bacterial cells independent of control of chromosomal DNA. Bacteriophages, because of their high number per cell, have very high copy numbers of their genome within the bacterial cell. Some plasmids may have only one or two copies per cell, whereas others may have 15 to 100 copies per cell. Their numbers can go even higher if we are able to learn an alien piece of DNA with bacteriophage or plasmid DNA. We can multiply its number equal to the copy number of the plasmid or bacteriophage. Vectors used as present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants. The following are the features that are required to facilitate cloning into a vector. First, origin of replication, which is ORI. This is a sequence from where replication starts, and any pieces of DNA when linked to this sequence can be made to replicate within the host cells. This sequence is also responsible for controlling the copy number of the linked DNA. So, if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. Second, selectable marker. In addition to ORI, the vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced into in a host bacterium. You will study the process in subsequent section. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloro, chloramephenol, phenocol, tetracycline, or kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Third, cloning sites. In order to link the alien, in order to link the alien DNA, the vector needs to have a very few, preferably single recognition site. Recognition sites for the commonly used restriction enzymes. Presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning. Figure 9.4. The, the ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at a BAMH1 site of tetracycline resistant gene in vector PBR332. The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on tetracycline containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on that containing tetracycline, but non-recombinants will grow on the medium containing both the antibiotics. In this case, one antibiotic resistance gene help in selecting the transformants, whereas the other antibiotic resistance gene gets infected due to insertion of alien DNA and help in selection of recombinants. Damn. Selection of recombinants due to inactivation of antibiotic is a cumbersome, which means a hard conversion procedure because it requires simultaneous plating on two plates having different antibiotics. Therefore, alternative selectable markers have been developed which differentiated recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta galactosides. This results into inactivation of the gene for synthesis of the enzyme, which is referred to as insertional inactivation. The presence of chromogenic substrate gives blue colored colonies if the plasmid is in the bacteria does not have an insert. Presence of insert results into insertional inactivation of the beta galactosides gene, and the clonines do not produce any color. These are identified as recombinant clones. Fourth, vectors for cloning genes in plants and animals. You may be surprised to know that we have learned the lesson of transforming genes into plants and animals from bacteria and viruses which have known this for ages. How to deliver
genes to transform eukaryotic cell and force them to do what the bacteria or viruses want. For example, Agrobacterium tuniferacensis, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA, to transfer normal plant cell into a tumor and direct these tumor cells to produce the chemicals required by pathogens. Similarly, retroviruses in animals have the ability to transform normal cells into cancerous cells. The better understanding of the art of delivering genes by pathogens in the eukaryotic host has generated knowledge to transform these tools of pathogens into useful vectors for delivering genes of interest to humans. The tumor-inducing TI plasmid of Agrobacterium tumefaciens have now been identified into cloning vector, which is no more pathogenic to the plants, but it is still able to use the mechanism to deliver genes of our interest into a variety of plants. Similarly, retroviruses have also been disarmed and now used to deliver desirable genes into animal cells. So, once a gene or DNA fragment has been ligated into a suitable vector, it is transferred into a bacterial, plant, or animal host where it multiplies. Competent host for transformation with recombinant DNA we will discuss in the next part. Thank you.